Welcome everyone to our second video on enzymes. Today we're going to apply some of our knowledge and we're going to look at the factors that affect enzyme rate of reaction. So the factors that impact enzymes are pH. So all enzymes will have an optimal pH that they act at. So a change in pH can alter the shape or the active site of an enzyme, which thus reduces its activity. Okay, so it's just some important terminology at the bottom of the slide. Anything that has a pH greater than 7 is basic. Okay, so the higher that number, the more basic or alkaline it is. A pH less than 7 is referred to as acidic, and the lower the number, the more acidic. Okay, so we can see here that we've got some enzymes in the body. We can see here that we've got pepsin, which is an enzyme found in the stomach. Pepsin has an optimal pH of 2, whereas the optimal pH for trypsin, which acts in the small intestine down here, the large intestine I should say, it has an optimal pH of 8. So depending on where those enzymes are found in the body, their optimal pH will vary. Another factor that influences the rate of reaction in enzymes is temperature. So all enzymes have an optimal temperature. Most of the enzymes in the human body have an optimal temperature of 37 because that's what our internal body temperature is. Okay, so if an enzyme's temperature or its environment becomes too high, the enzyme will denature. When something denatures, we have a permanent shape change from broken bonds. This is irreparable and the enzyme will be inactive. If we have a low temperature, enzymes don't work properly, but they can still begin to work properly as the temperature gets closer towards its optimum. Essentially what's happening here is as we lower the temperature, there are fewer particles colliding and less chance of a reaction. So high temperature, denature, enzyme's gone, we can't use it anymore. Low temperature, the enzyme just will work very slowly, and as the temperature gets closer to the optimum, it will start to work at its appropriate rate. It doesn't get damaged or destroyed. Okay, so this diagram here is showing you a normal protein or a normal enzyme and what happens after denaturation. So after we have that heat, the bonds will break and that protein will no longer function as it should because its shape has changed so much. Okay. Another factor that impacts enzyme reaction rate is enzyme concentration. So let's think about this logically. If we've got lots and lots and lots of enzyme, we would expect that our rate of reaction would increase up to a point where we haven't got any other substrate molecules that will fit into those enzymes, so it will continue to occur at a steady rate. Okay? And we can see a little graph here which shows that. So as we get more enzymes, the reaction goes quicker, up to a point where every enzyme has got a substrate molecule, therefore the reaction will keep occurring at the same rate. Factors that affect the rate of reaction in enzymes, I suppose the second one here is substrate concentration. So this time we're thinking about the thing that the enzyme is acting on. Okay, so as we get more substrate, we would expect the reaction rate to increase up to a point where every single substrate molecule has got an enzyme, or we say that the enzyme is saturated. We've got every enzyme has got a substrate, so therefore the reaction will plateau out and level because all of the available enzymes are being used. We haven't got any more enzymes to help those substrate molecules. So we say that that point there where the graph levels is called the point of saturation, where all enzymes are occupied. When a substrate gets too high, as I just said, the enzyme may become inhibited. So there's so much substrate that the reaction just can't proceed any quicker. Okay, there are some instances where we see enzyme inhibition. So enzymes can become inhibited when other molecules compete for the normal substrate area on the active site. So other molecules stop the normal reaction because they block the active site. So we can see here the normal substrate, normal enzyme fits straight into its active site. We can sometimes come across instances where we have what we call competitive inhibitors, which block the active site. We can also have non-competitive inhibitors, which bind to the enzyme anywhere else except for the active site. So I say they bind to an allosteric site. Allosteric means any site except the active site. Okay, so competitive blocks the active site, non-competitive doesn't. It just attaches, it causes the active site to just change shape slightly, and that allows the substrate molecule, or prevents the substrate molecule from binding. Okay, another diagram here to demonstrate this idea of competitive and non-competitive inhibitors. Some enzyme inhibition can be reversible and some inhibition is irreversible. So some examples of irreversible inhibition comes about when we've got toxins or cyanides as an example here. So this is an example of something that can't be reversed and it can result in death. It's quite dangerous. Okay, reversible is when our inhibition can be undone, and most forms of competitive and non-competitive inhibition are reversible. So most molecules that our body makes to act as competitive or non-competitive inhibitors can be removed through a feedback loop. Okay, we can see here the difference in rate of reaction. When there's no inhibitor present, the reaction is quicker, and when we've got a competitive inhibitor, still works, but the reaction is at a slower rate. Okay, this is an example of when inhibition can be really important. Okay, so it's often used as part of negative feedback loops. So that's a useful diagram to add to your notes. This slide provides an overview of the factors which affect the rate of reaction in enzymes and their effect on the rate and the reason. This is a super duper important slide. Please add that to your notes because that will allow you to be able to help you to answer some application type questions when we get there. Okay, now we've got a question for you to answer on Edpuzzle. Okay, so the answer to that question was C. Cofactors and coenzymes. This next slides provide you with a little bit of an overview and application of enzymes. So enzymes sometimes require cofactors or coenzymes in order to function properly. So cofactors are small inorganic substances that must be present in order for the enzyme to work and catalyze the reaction. A coenzyme are small organic substances that must be present in order for the enzyme to work. So cofactors are inorganic. They're usually ions like magnesium. Okay, coenzymes are organic substances, and anything organic contains carbon. That's how you know if it's organic or not. So if something contains carbon, it's organic. If something doesn't contain carbon, it's inorganic. These are some examples of some cofactors and what enzymes require them in order to function. Okay, this slide provides you with some examples, ones that I want to draw some attention to here, okay, are down in the coenzyme section. So coenzymes play a really critical role in transferring functional groups in enzyme catalyzed reactions. So some coenzymes that we're going to learn about down the track are NAD, NADP, and ATP. All of those molecules carry energy in some way. So we'll learn a little bit about them later on. What I need you to know today is that coenzymes are important organic molecules that transfer energy or functional groups. Okay. Coenzymes are constantly recycled and reused in chemical reactions. There are a number of special coenzymes, and often these coenzymes can come in a loaded form or an unloaded form. We will learn about these more when we look at photosynthesis and respiration. when we learn about how in photosynthesis NADP carries hydrogen molecules, and in respiration NAD or FAD carry hydrogen molecules. There we go. There's an example here of what NAD does. We'll also look really closely at ATP and its critical role in transferring energy when we learn about respiration and photosynthesis.